Welcome back to Chapter 5 of Microbiology. Today we're going to be covering a little more on viruses and other non-cellular infectious agents. First, we'll be looking at how they multiply, how they infect their hosts, and find further hosts. Then we'll look at how we can study them in the lab. Other non-cellular infectious agents, such as prions, and their role in human health. Right, so our first section will be on how viruses multiply. So just a quick reminder, viruses are minute or very small parasites that can hijack the genetic material and genetic machinery of cells. They're very small because you can see in the background here, green is the host cell. And then the gold is the bird flu virus here. So much smaller, many of them can fit within the host. And as we talked about last time, they're non-living. They can't reproduce on their own. They require a host. And to do that, they need to hijack the ribosomes or the transcription or translation machinery. There are a couple of steps that the virus will go through to multiply, especially in animals. The general stages of a viral life cycle are absorption, penetration and uncoating, synthesis, assembly, and release. So if you look over here on the right, we have our viral particle here, and it has its lovely enveloped spikes out here. They're going to try to find a receptor. And if it's a match fit there, a perfect lock and key fit, then it's invited into its host. So first it's going to have to dock and be absorbed into it, penetrate and get uncoated and get any of its genetic material out. Then that genetic material can be used to synthesize all the components of the virus that can then be assembled, and then they can be released to find new hosts. Your book has this great figure here, a whole page figure showing the stages in detail. We first start with the absorption, where it's going to dock via its spike protein. So maybe you heard with COVID-19 that it had certain spike proteins that docked very well with human cells. Okay, so it would kind of lock in there. And then second step, it can penetrate and uncoat it. So it's kind of like phagocytosis in reverse. So it's kind of being sucked in, making a vesicle around it with the host membrane. Then 3, it's going to be released out. Any of its RNA or DNA will be released into the cytoplasm, and it can be used for replication of its genome, copying more copies of the RNA or the DNA, and then also production of any of the protein components. So things like new spike proteins, capsomeres, maybe if it has one special enzyme, any of that is going to get made in step 3. And then once we have all of the nuts and bolts basically of the new virus, all of our components to, I don't know, our little IKEA assembly here, then we're going to put them all together. In step 4, assemble all the capsomeres, put the genetic material inside, and then it's ready to be released outside of the host cell to go and propagate its genetic material and find further hosts. Okay, so again, the main steps are absorption, where it docks with its host, penetration and uncoating, where it gets kind of reverse phagocytosed, kind of sucked in here. And then it's going to be uncoated. And then we're going to have synthesis of its proteins and its genetic material. Going to put it all together in assembly and then release it into the environment. Okay, so we're going to go through each of those in detail. Absorption is our first one. Viruses can only invade cells that have the specific docking sites, the specific molecules on the cell surface. Cells that lack these receptors are therefore going to be resistant. They cannot be infected. This virus can't get in. Therefore, it can't use its machinery and it can't bother it. This means that there's a limit to the types of hosts that it can infect, and this is known as host range. So some examples of host range could be things like hepatitis B can only infect human liver cells. It's not going to infect human brain cells. It's not going to infect bat cells. It's not going to infect skin cells. It's only one specific species, humans, and a very specific cell type. Another example would be poliovirus. It can infect intestinal and nerve cells of primates. So this one has a little bit broader host range, meaning it could infect chimpanzees or orangutans and humans, but only intestinal and nerve cells. Rabies virus can infect pretty much any type of cell in all mammals. So this one of our three examples has the widest host range. The next step is penetration and uncoating. So once it has docked with those receptors, it's ready to be taken in. The host membrane can be penetrated either by the whole virus, so the whole thing, all the capsomeres, the capsid, all of the spike proteins, the whole thing could be taken in, or it could just basically dump its nucleic acids, its DNA or RNA inside of the host cell. Endocytosis, or the taking in of the virus through invagination or engulfment, is often done in this step. So that's when it's going to be kind of sucking in the virus. Once it's brought into the host cell, enzymes inside of it may help break down the viral capsid, that kind of outside coating, to reveal the genetic material that's going to be used to make more copies of the virus. Can also have direct fusion. So in this one, the viral envelope will fuse with the host envelope, and it will just dump the nucleocapsid inside. So if we look at that a little bit more, you can either have the docking and engulfment, and the whole thing is brought in inside of a vesicle that can then break it down and get the genetic material out. Or we can have attachment and the membranes will fuse and just the nucleocapsid will be released inside. But either way, we're getting that genetic material into the host cell. Our next step after the genetic material has been brought into the host cell is synthesis. And this is going to involve replication or copying of the genetic material, and also protein production to further make the actual structural components of the virus. Most DNA viruses will go, their genetic material, that DNA will go into the nucleus, and then there the DNA will be replicated and assembled. On the flip side, most RNA viruses will be replicated and assembled in the cytoplasm. These locations are very similar to, if you think back to Bio 1, to replication and transcription. If we're going to copy DNA, that's going to be kind of more affiliated with nucleus, whereas RNA and transcription and translation is more affiliated with the cytoplasm. The double-stranded DNA viruses have two stages to their synthesis. The DNA will enter the nucleus where genes coding for proteins needed are there, and they're going to make the viral DNA and make it into RNA. Then it's going to be sent to the cytoplasm for translation, and the viral DNA can be replicated. The capsid proteins and other structures will be transcribed and translated. So they're going to go from DNA to RNA, transcribed, and then from RNA to protein to be translated. There's many steps when we're going from the DNA. RNA kind of takes a little bit more of a shortcut. So once the genome has been replicated and all of the necessary proteins have been produced, then it's ready to put together the whole virus. Basically, it has manufactured all the nuts and bolts and all the pieces it needs. And once they're all there, they're ready to be put together. Once it's been assembled, it's ready for release, where it can be dumped outside of the host cell, set free to go find more cells to infect. The number of viruses produced is dependent on the size of the virus, as well as how healthy the host cell is. So the smaller the virus, the more of them you're going to make, as well as healthier host cells will make more viruses than sickly ones. You can see in the background here, the number of viruses shown out here in kind of magenta to one host cell in the middle. Just to give you some context on the numbers, polioviruses can be made in the 100,000 range. So one host cell can make 100,000 viral particles. And each one of those could find their own host and infect it. So you can see how a viral infection could get really out of hand really quickly. But then again, not all of those viral particles will find a host. So not all 100,000 of them are going to find a host, but even if only a small number do, that is potential for rapid proliferation. So even if only, I don't know, 1 out of every 100 finds a host, we're still at 1,000 more cells infected from just that one starting cell. Okay, so this shows the release here. We have our host cell, we have the RNA that's been copied, the capsid proteins that have been made, and then they can be packaged into the budding viral particle with its spikes on the outside here. And now it's a free infectious viron ready to go find a new host.